Alright, I'm going to show you what a mud fossil is, and I'm going to explain the process and how they're created. Now, this is a head of a bone of some form of an animal, I don't know what kind. Now, it would have been a long bone, I don't know how long it would have gone for, but the internal structure here is still contains the organic matter, which is the iron and with the blood and so forth inside here. And there's a lot of articulation, a little tiny canaliculus they call them, and then there's also the fascia that covers it. This is the cartilage in this area and over here. Normal bones don't have that. They're stripped from microorganisms. This right here is where the ligament attaches, and those little fibrils insert in there, and that is literally on cartilage. When they find the bones in the sandy soils and the porous soils where oxygen can get down there and microbes and eat all that stuff, they end up with bones that are really stripped right down to nothing. You can see here, I believe these are the blood supplies, or there's some little fibers that come through there. And if you look on living bone, it's all here. It's all exactly here. And I will show you a structure that looks like that. But when they find them in the ground, in the sandy soils, they're torn down to shreds, down to these, all the literally just the mineral. Now, the bone I showed you has a periosteum on the outside. It has all kinds of blood supplies running up through here. It has fascia that coats it, and the fascia I will show you. Now, normally this would, in the cartilage, it had all that stuff. This would have is attached over here somehow. I forget how it goes, but this is cartilage. That goes away because animals eat it, and you end up with these kind of bones. And that's the kind of bones they're looking for, and that's not kind of bones that you find in mud fossils. Mud fossils have the entire structure of the bone totally articulated. Now, if you look closely, there's some separations here where these little ears would fall off. And you also, if you look closely, you'll see this is a ligament attachment. One was here, and one was over here. They attach, boom, boom, like this. And this is, those might have been the tendon attachments, ligaments. I think they call them the ligaments. But this one here is ligament between bone to bone. And these are the little tops here. This whole top is cartilage. And now here's one that's worn down a little more. And you can see that the cartilage on the top, that the other one has cartilage that comes all the way around here, and this one is worn down, and even the holes and all that stuff are worn down. Now, something that is, I'm going to show you some spectacular stuff. This right here is what they call an apical tuft. And that is on the tip of a distal phalanges of probably all kinds of animals, but humans have them, and they're right on the very tip of here, and I will show you that in a second, what an actual distal tuft looks like. But that's it right there. And that also has fascia on it. If you looked at this very carefully, you could see that fascia. It's a fabric that coats all the bones and organs in your body. This is a better representation of fascia. And if you look at this carefully, you're going to see a lower investment of fascia, an upper investment of fascia where it's snapped off. And that, I believe, is a lung of some form of an animal. Now, it also has a depression in the back where the heart would sit. It also has these structures over here that are patterns that appear in an actual human lung. Now, I'm not saying this is human, I'm just saying maybe all lungs are the same. But they have this fascia implantation at the bottom. It invests the lungs and holds it from flopping around in your body. And this, believe it or not, is a fossilized investment. All right? This would, right there, if you can look carefully, I don't know if you're going to be able to see this, But that is what they call fascia investments. And this is literally petrified flesh. And you actually can see fibrins running through here, the elastins, the little wavy black elastin fibers. I don't know if you'll be able to see them or not, but they're in there. I have all the documentation on this. Now, in addition to that, as crazy as it seems, this is what is a distal phalanges, and this actually has, you can see the pattern of this apical tuft in here, if you look at it with the microscope. But this is what they call carbon bone staining, or it's also iron, can be leached through. Now if you look very carefully, you're going to see a pattern. I don't know if you're going to be able to see that, but it comes down and steps over like this. And out here is that apical tuft. This particular tip of whatever it was from is partially degraded. Now notice on the right here, you see that tube? That's a vein. This side is the artery. And these two holes here are where the artery blows out the blood on degasification when it goes out. There's also two holes on the end. You see this side? This side has the holes that blew out. This is the side that blew out. That's also the artery side. Veins don't blow out. That's the vein side. And these down here are the veins. And the reason they didn't blow out is because that little tip on the top is what's called a venal clamp. And they clamp and hold, they don't let the blood flow. This side they blow out like a firecracker. All of that signs are here. Now, that is what's, hold on one second. Okay, once again, I'm not claiming this is a human. I'm saying, because obviously not a human. That thing would be, if it was human, it's around 60 feet tall. I calculated it out. But anyway, you see how this splits off? One runs up this side, one runs up the other side of the other finger. One side is a drain back to the heart and lungs. The other side is your artery that pumps the blood down. So if you see on here, it's an identical pattern, because we're only looking at this very tip of the finger. And at the bottom there's two entrances. At the side there's two, at the end there's two, and at the other side there's two. And one of them brings the blood in, and the other side lets it come back. Whoops, this is about tendons. But anyway, the idea of the veins and the arteries is one side doesn't blow out because it has internal clamps, and I have seen this many times, and I have evidence of this where they actually leaked blood backwards. Now, this, I am saying, is the same as this, and it's probably from the same creature. However, it is not as deteriorated. Now, if you notice this here, this cut, if you can see that, those triangles are where the areolar tissue, the outside connective tissue, connects to the underlayment, and you can see the same pattern there. That is exactly the same. And this, if all that areolar tissue, which is the holes, they call it areolar tissue because it has little tiny holes in it, if that had deteriorated away, you would be at this point. And this, I believe, is the same as this. This is the apical tuft of this distal phalanges, and this would be right on there. The only thing that the bone is, this ball, I remember I skipped over and I hit that tendon. There's a little ball on there, and that's these little balls. And that's what inserts into the tuberosities of these tips of your fingers and of your toes and everything else. And I mean, I'm not saying this is human. It's obviously not human, but it is what it is. Now, this stuff came deep, deep, deep from the earth. If you look over here, this is a stone. It's virtual block. And that is what happens deep in the earth. They turn somewhat liquid and they sculpt into different shapes. And that is a squared off ultramafic, or he might even be tectonite, but it's deep, deep in the earth. I have a whole bunch of stuff to show, but and I'm going to do videos on all of the different parts because it's a not understood process and it's not accepted. And I show this stuff to, I mean, I can't see how you could say that is not a bone. It's not. If you look at and I'm going to show you the actual bone of a living bone, what it should look like in its entirety as an organ, because this is really, all of these things are organs. This is bone that they find in the ground. That's a stripped down bone. That's just scraps. There's all kinds of articulation on a living bone. There's cartilage, there's canaliculus, there's all kinds of vessels and ligaments and all kinds of things attached. And I will show you all, I'm not even going to show you that stuff now because it's just it's too obvious that that's what this is. And there's never ever been a bone found or reported in Connecticut because they won't accept them, and we're right down the street from Dinosaur State Park. There's all kinds of dinosaur things around there. And I think I have a lot of, I don't know what they are, they're pretty big to me. There's small ones, there's big ones. But that's what I have. And but this black carbon staining I showed you before, that's what happens when carbon leaches. You see that? That's the pattern of the bristles inside the wood. They leach. It's just a characteristic of carbon. And carbon is the last thing to go under sublimation. Now, I have transitional fossils that run from being almost perfectly, totally not touched, all the way down to just totally crystallized, like this right here. You see that crystal? I believe that at one time was a lung. I know you're going to say, oh, this guy's crazy. Well, let's find out. This, I believe, at one time was a lung of some kind of an animal, and I don't know what kind. But what happens is as they sublimate and all of the organics turn into methane and bitumens and everything, you end up with metals and iron and all that business in here, and it starts to create crystallization, and crystals propagate from the base little seeds, the metals that are left over in the body after partially transitioning into fossil fuels. Anyway, I need collaborators. I need somebody to test this stuff. Nobody will test it. I've even offered to pay to have things tested, and nobody will do it. So I need somebody to come along and help me along here. It's rspurr, rspurr, @gmail.com. And I need like somebody to do the tests on this stuff. So, and I do need stone saws, and I need some excavation, because believe it or not, this stuff here, this is tiny. I have huge things out there, huge. I mean, that came off of something that was a big bone, and I have the big bones out there. I have a couple of them sticking out of the ground right now, but I can't get them on the ground. All right, so somebody get a hold of me. I'd like to get this thing done, because if I go, this stuff goes with me. Nobody's paying attention to it, and I've got to have it paid attention to, or I will die an unhappy person. All right? Don't let that happen. Thank you. Okay, I'm not making any claims this is a human, but you can see that's the distal tuft. It's exactly the same structure, and these little tiny holes and holes there are where the little balls of the tendons attach. They're called enthesis points, and I'll show you that in a second. Alright, if you recall, right there is where a ligament, or they attach the same way, ligaments and tendons. They have those little fibrils, the little tendrils that go right down deep into the substructure, and they actually, there's other things they call, I think they call them Sharpie's fibers, that eventually dig into the bone a little bit. But this is primarily in the cartilage. Now, this is what it looks like in a normal tendon. It comes down, you have fascia, you have all kinds of sheathing around there, synovial sheaths, you have 3 different separations of them so that they can do their little pulling back and forth thing. And as it comes down further and further and further, you end up with these tiny, tiny, tiny little microfibrils. And those are what go into the cartilage. And I think they're not even sure how these things attach, because it's very difficult to see in living tissue. It's sort of a globular thing, but here it's pretty apparent. There's a cavity, and in that cavity there's a ball, and coming out of that ball there's fibrils that go into the deeper structure of the cartilage and into the bone, it looks like. Alright, this is a stripped bone, but you can see where the two ends here, that fed blood supply, I believe, from bone to bone. There's also what they call foramens, and those have, inside there's a vein and an artery run in there, and they do the same thing there. One bleeds and one doesn't. I'll show you that in a second too. Alright, this is that fascia. You see the structure of it. Now this is one of those bone foramen. That cavity is where blood vessels go in there and come back. Now this is when I took that red scab halfway off, and that white structure underneath, I believe, is what's called fibrin. And fibrin is the clotting fabric that clots blood. And then when I took that out completely, let me see if I can show you this a little better. There's two holes. Oops. Anyway, there's two holes right down. There's one here, and there's one right here. This one didn't bleed back because it's the artery, and they don't have any blood left in them. They purge. This one, you see it ran down, ran right down here, and that's what caused that clot. Anyway. Okay, now I'm going to explain to you the process of mud fossilization, and it relates to micro-size platy little polar silicates that seal off unfortunate mudslide creatures that got captured in mudslides, completely encapsulated in the tiniest of micro platy polar silicates. They're clays, and they're continuously wet, and they encapsulate and keep any air and oxygen away from the thing, and then because they're wet, they create what's called spontaneous potentials in the earth. And in these wet environments, there's little tiny microcurrents that run through the earth. Now, the ionic nature of flesh is that it will actually literally plate from the molecular little polar, which means they have a positive and negative end crystals. They're flat, they're like little plates, and they migrate towards these ionic fleshy parts, and they plate them just like you plate cheap jewelry, or you would shingle a house. And they keep getting thicker and thicker on the surface, and it encapsulates it more and more, and the internal structure is literally isolated and can't deteriorate. And this right here, if you see this tissue, that is chicken tissue that I put under 12 volts inside of literal sand, just sand, and it eventually plated that chicken tissue just like that. That stuff doesn't fall off. It's adhered to the surface. It's been rubbed off, and it doesn't rub off. I'm doing some more right now, and they've been in there for about 3 or 4 months. It takes a long, long time. Anyway, that's what happens. And when they encapsulate, they literally keep the tissue, from the internal organisms, everything inside, from being destroyed. And eventually that plating becomes thicker and thicker and thicker, and less smaller molecules get through there. And I believe the result is that OH, being one of the smallest molecules, eventually goes inside, and hydroxide causes the bones to deteriorate. It interacts with the phosphorus and the calcium, and it causes the actual bones to turn into, well, I have some examples of it, but anyway, I believe that's why you don't see the literal bone structure inside of mud fossils. So there's an answer for everything. It's just got to be checked out, and I need people to do the tests. I can't afford it, and I don't have, nobody will do them for me. And even if I could afford it, I haven't found anybody that's willing. All right, so we need to get together and do this.